some accessory structures of the eye. This slide is not in your PowerPoint, but one of your objectives is the functions of your muscles, okay? So, of this picture, you need to know the name and the action. You don't have to know the controlling cranial nerve, okay? And they come in pairs. So you have the lateral rectus and the medial rectus. They move the eye laterally or medially, okay? They make sense, okay? Then you have the superior rectus and the inferior rectus, okay? These do partially what you think. So superior rectus does elevate the eye. The inferior rectus does depress the eye. But they also are responsible for turning the eye medially. So if you see a superior inferior rectus, add medial to the depress or elevate. Okay? Then finally, we have the two obliques. Now, obliques mean they run at weird angles, which means they do weird things. So our superior oblique depresses the eye. So it's the muscle above the eye, but he actually pulls it down. Okay? Again, it just has to do with the weird angle it runs at. The inferior oblique is going to elevate the eye. Again, it sounds opposite of what you would think it does. And then both of them turn the eye laterally. Okay? So just make sure you know your actions with your muscles. Right? Okie dokie. Then we will go on. Okay. For your first homeostatic imbalances for the eye and vision. Strabismus. Also, you might have heard it as lazy eye. Okay, so this is where you have a congenital weakness of your external eye muscles, and it prevents at least one of your eyes from staring straight ahead. So one set of muscles are going to be stronger than the other set, and the stronger muscles are going to pull the eye that direction. So if you have this person right here, and their affected eye is this one right here. Okay, and it is pulling medially. So the stronger muscle is going to be the medial muscle. So the medial rectus would be stronger. Okay, the muscle that needs to work to pull it back this way to get it straight and center, the lateral rectus is not as strong. Okay, that ends up having a lazy eye, a movement of the eye. Okay, so treatments. You do eye exercises and you have the patient wear an eye patch on their good eye. So they would wear an eye patch and cover this one up. So now that eye isn't doing anything, and in order for that person to see, they have to work that lateral rectus to make the eye point straight so they actually can see what's going on around them. Okay? So it's hard work. It takes a lot of time. It takes some patience, but it's possible to do. Okay? You can also do some surgery that helps correct it if the eye patch itself doesn't work. Okay? All right. Okay. I am skipping that one because we are a little behind the other class. Okay? So, now, part 2 of chapter 15. We are now going to get into the anatomy of the actual eyeball itself. Okay, and the anatomy of the eye that is responsible for vision. So vision is the perception of light that we see around us. And light itself is electromagnetic radiation. It's found on the electromagnetic spectrum, and it's measured in wavelengths. So if we've got our electromagnetic spectrum down here, the same type of waves are found in radio waves and microwaves and X-rays and gamma rays. But what they do depends on their wavelengths, how big the waves are. Visible light, the light that your receptors in your eye are able to pick up, is found right here, just in a small portion of the electromagnetic spectrum. Okay? And then what color you see is dependent on what the wavelength is within that small window. Now, cool fact. Most of the solar radiation that reaches the Earth falls in the 400 to 750 nanometer range, which coincidentally matches up with the receptors that are in your eyeball. So, the receptors in your eyeball So this slide is a review of accessory structures. So conjunctiva is that clear covering over the eye. What's it called when you have inflammation of your conjunctiva? Conjunctivitis, right? Yes. Yes. It depends on what the source of light is for the other planet. If it's still in the solar system and the sun is what reaches it, you'd still be able to see. Now, the brightness would be different depending on where you are in the solar system and the atmosphere of that planet, right? Because the atmosphere helps block some rays. But if you went to another solar system that was lit by a different star, you may have a different set of wavelengths that reach the planet. Yep. Yeah. Okay. I'm not going to go off on tangents. We're going to stay on topic. Okay, so conjunctivitis. Okay, next is your lacrimal apparatus. What does your lacrimal apparatus produce? Tears. That's right. And your extrinsic eye muscles are responsible for moving your eyeballs around. Okay, so now, when we look at the eyeball itself, we can separate the eyeball into 3 different tunics, or 3 different layers, okay? So we're going to talk about some of these things, and then I'm going to have a picture up here for you, okay? So the tunics, we have 3 layers. The outermost tunic, the tunic that you see when you look at somebody's eye, is called tunica fibrosa. And I kept talking last class about the cow eye dissection, and they looked at me funny. Do you guys dissect cow eyes in lab? No? Okay, so I will try not to reference the cow eye as much this class so you guys won't look at me funny. Okay, but on the outside of the eye, we have this fibrous tunic. It is really, really thick. Okay, it is made up of the sclera, which is the white part of your eye, and the transparent cornea, which covers the very, very front. Okay, this is super thick. If you do a cow eye dissection, I told you I wasn't going to reference it, but it just comes up. If you do a cow eye dissection, in order to get through this fibrous tunic, you have to stab it with your scalpel and then saw through it. It's super thick. Okay, super thick. Okay, the second tunic is the tunica vasculosa. So it's a vascular layer. There's going to be blood vessels there. Okay, it has a layer to keep the inside of the eye dark, and that dark layer is called the choroid. It's a dark brown membrane, so that when light comes into the eye, it doesn't just bounce around. Okay, it goes to the retina, it gets absorbed, and then that dark layer absorbs the rest of the light. Okay, it doesn't just bounce around on the inside. However, there are some animals, like the cow, that that also have a layer on the inside that's shiny and shimmery. And that shiny, shimmery layer helps light, when it comes in, bounce around. And that's for animals that do a lot of outside work. That's not a way to say it. But for animals that live outside and are around at night. That's what I'm trying to say. Okay, so when light comes in, if it's in low amounts, it's going to come in and it's going to bounce around the inside of their eye because of that shiny layer, and they're actually going to be able to pick up light, even if it's just a little bit of light. So nocturnal animals have this layer. Yeah? Yes. So what happens for animals that see infrared? Our cones in our eye, we're going to get to this photoreceptors later, but they are specialized to pick up red, green, and blue, which are visible light spectrum. They have photoreceptors that pick up a different wavelength, and they pick up that wavelength that's in the infrared spectrum. They pick up. So theoretically, there could be an animal somewhere that we don't know about, theoretically, that has receptors to pick up radio waves. I mean, they're the same type of wave. They're just a different wavelength. Okay, so they may have photoreceptors that pick up all those different wavelengths. Guys, so I wasn't really distracted. Do any of you guys listen to podcasts? Okay, a couple of you guys. There's a podcast called Radiolab that is awesome. It's like some good science there. And one of their stories was about photoreceptors in different animals. And there's like this shrimp who has tons of cones. Like we have 3. We have a red, green, and blue. That's it. But these shrimp can pick up wavelengths that are in the infrared and in the ultraviolet range, and then everything in between. So they have like really good vision, and they're shrimp. Anyway, what was I talking about? Tunica vascularis. Yeah. Yes, like he is super powerful and crazy. Yeah. Yeah. All right. Tunica vascularis. So we've got this dark layer that keeps the light from bouncing around. The refractive one, though, like you see in a cow, you also may see this like in deer eyes. So when you're riding down the road and your headlights hit them and it looks like their eyes are glowing, it's because the light is hitting that reflective surface and bouncing back at you. Okay? All right, but we don't have that. Red eye, though, like when you take a picture, again, I'm getting myself. When you have a flash on the camera, the light goes into their eye and bounces off this tunica vasculosa, which has blood in it, and that red is what bounces back. So you're seeing the back of your eye. Yeah.
What was the refractive layer called?
It has a name, but I can't remember it. Do you guys remember? Have you guys done the cow dissection? No. Okay. It may come to me later, but I don't remember right now. Sorry. Okay. So also in that tunic, we have a ciliary body that forms a muscular ring around the lens. So the ciliary body is going to constrict or tighten up, which will change the shape of the lens, which will help focus light on the retina. Okay. We also have the iris, and the iris's job is to control the size of the pupil. Okay, to constrict it and make it smaller, to bigger and dilate it. He contains chromatophores with varying quantities of pigment. This is what gives your eye the pretty colors. The lighter your eyes are, the less of this pigment you have. Okay, so you have a lighter pigment, which is going to allow more light to get into your eye because the light can go through your iris, okay, if you have a light color. Whereas if you have a dark colored eye, like a dark brown, that blocks more light from getting into your eye. So if you have a lighter colored eye, you may be more sensitive to light because when you go outside, your light's coming in your pupil, but it's also getting in the iris area as well. So you let more light in. Okay? Did you have a question? Okay, good. All right, and then finally, the tunica interna is the most inside layer, and it's the retina. Okay? So now let's look at a picture of all these. Morgan, you emailed me about the diagram, right? No, that was you. Somebody emailed me about the diagram. Which diagram is it supposed to be? Oh. This is the diagram you need to know for your objectives. Sorry, Morgan, I thought it was you. Okay, so in your objectives, there is one that says know these parts on the diagram from lecture. Here is your diagram from lecture. Okay? There are 3 things on this diagram that are marked out by the blue boxes that you do not have to know. Everything else, you do. Okay? So let's talk about them real quick. You've got your 3 tunics up here in the corner, so let's go through them. Your tunica fibrosa is the most external. So it includes the sclera, this white part, and it includes the cornea, the clear part on top. That is the tunica fibrosa. Okay? Your next layer, the tunica vasculosa, is going to be the choroid, which is this dark layer right here, which in reality is kind of a brownish black color. You've got your ciliary bodies up here, which are muscles. The ciliary body holds onto suspensory ligaments right here, and that holds the lens. Okay. It also includes the iris, which is up here, and the pupil, which is the opening. Okay. And then finally, the tunica interna is the most inside layer, which is your retina that goes all the way around the back. And in this picture, it is yellow. but in reality it's more of an orangey color. Some might say it's even a crimson orange color. Okay, so things on the retina that you need to know. And there'll be a slide that like talks about these, but I'll go ahead and tell you while we're looking at the picture. You see this little dot back here? That's the macula. It's macula lutea, but usually you just hear it called the macula. Okay, and inside the macula is the fovea centralis. There's a little dot in there. This is the area of the highest visual acuity. So if you are trying to focus on something and get it really clear, you want it to focus on that spot. That's where you have the most cones. Okay? Then we also have, so the fovea centralis and the macula is the highest visual acuity. Then you have your blind spot, okay, or your optic disc. There are no rods or cones on your optic disc, okay, so you have no vision at that spot. And the reason you have no vision at that spot is because that's where the optic nerve exits the eyeball. Okay? So there's no room for rods and cones there. Alrighty, and I think the last thing I need to do is talk about the anterior segment and the posterior segment. So the anterior segment is going to be the area in front of the lens. Okay? Of this anterior segment, it doesn't have it labeled on the picture, but it'll be important for the next slide. There's an anterior chamber, which is in front of the iris, and a posterior chamber behind the iris. And it is filled with aqueous humor. And aqueous humor is very, very watery. So again, if you do a cow eye dissection and you cut it open, this aqueous humor just looks like water. It just runs out. No biggie. Then in the back, in this posterior segment, this area is filled with vitreous humor. Now, once you've gotten your scalpel through the sclera and you sawed your way through, and you're into the posterior chamber, or posterior segment, this vitreous humor looks like jello that you've left out in the sun too long. So it's kind of sour, kind of that in-between stage. Okay, but the purpose of both of these humors is to maintain the shape of the eyeball. So we're filling up the inside of the eye with something to help it push out, to help it maintain the shape. Okay? All right, so I think those are all the parts. Did I miss anything? Yeah.
What causes someone's eye to be like more than one color?
Okay, so that has to do more with genetics, and it's the genetics controlling the pigments of the iris. So you have people who have multicolored, both of their eyes are like a hazel color, where it can be a bluish, greenish, brownish, and it depends on kind of the day and what you see. There are other people who have like one eye one color, one eye the other, which is also, it's just a genetic thing. All right. And the genetics of eye color is similar to like the genetics of hair color. There's a lot of stuff going on. It's not just something is dominant over the other, and that's what you get when the baby pops out. Pretty intense. Okay, moving right along, the optical apparatus. So when we talk about the optical apparatus of the eye, we are talking about the path that the light is traveling through the eyeball. Okay, so it emits and refracts light rays, so it's going to let the light through, and it focuses the light on the retina. Okay, so here at the bottom of the slide are things that I already told you with the diagram. Okay, but the lens is connected to the ciliary body by the suspensory ligament, and those structures can help change the shape of the lens, which allows you to focus on things either far away or up close. We are going to talk about this more in detail in a second. Okay, right now we're just looking at anatomy. And then the anterior and posterior chambers of the anterior segment is filled with aqueous humor. And then behind the lens, the posterior chamber, or the posterior cavity, is filled with vitreous humor. Okay? So that's just words to go along with what we just talked about. All right. Questions? Okay. All right, the pupil. So we need to talk about the opening and closing of the pupil. We talked about this some with our autonomic nervous system, with sympathetic versus parasympathetic. Remember that that's going to be on the test a Okay? Which one of these two, I realize it's on the picture, but which one of these two causes dilation? Sympathetic. Why do you want dilation for the sympathetic nervous system? So you can see more. You see things that are farther away, you let more light in. Okay, so sympathetic is going to control a muscle called the dilator pupillae, which causes the pupil to dilate. Okay? The parasympathetic does the opposite. It causes constriction. But he's going to use a different muscle. He uses sphincter pupillae, okay, which contracts and causes the pupil to get smaller. Okay? So we talked about this in terms of seeing things far away and seeing things up close. What about light levels? When do you want to have your pupil constrict? When it's really bright, right? You want to limit the amount of light that comes in. And then you want dilation when it gets dark, right? Okay. And you guys have experienced this. You've gone into a dark room from the bright outside. When it was bright outside, your pupils were nice and constricted. You go into a dark room and then you can't see anything because your pupils are still constricted. Or you do the opposite. You go from a dark room where your pupils were big and dilated, and then you go out in the sunshine, and the sunshine burns your retina because your pupils are still dilated. Okay? And that's why we have this constriction, so that we don't burn our retinas. Okay? It's why you don't stare at the sunshine. It'll burn your retina. It happens. It's true. Okay. Yeah.
What happens if one of those muscles doesn't work? Like, does that make sense?
Yeah. So if one of those muscles doesn't work, then that indicates that you have some type of nervous system problem. The pupillary light reflex, which we're going to talk about in a little bit, where they shine a light in your eye to see if your pupil constricts, is like one of the tests to see if you have brain stem control, to see if you're still alive. So, yeah. Okay. So if it doesn't work, you're probably dead. Okay. All right. The retina in the back of your eye. Okay. So we're going to talk about these layers. Now remember, our light is coming in our eyeball this way. And it's going to hit the retina, which is covering the whole inside of the eyeball. Okay, so that's where we are right now. Okay, the retina has a couple different layers. And we can see it back here. And then we'll zoom in. But this is like an actual microscopic picture. Okay? So back here in the back, we see the choroid, which is that dark pigmented layer of the tunica vascularis, which prevents light from scattering inside. And then right in front of that is the pigmented layer of the retina. You can see this layer of nuclei right there. Okay? So the most posterior layer of the retina is that pigmented epithelium. Again, it doesn't do anything as far as absorbing or taking up light information and sending it to the brain. All it's there for is to absorb any extra light, okay, to keep it from scattering. The rest of this, though, these other three layers that we're going to talk about, make up the neural apparatus. Okay? And so that neural apparatus is important for picking up photo information and sending it to the optic nerve. Okay? So let's talk about those three layers, the neural apparatus. Okay? We are going to work our way from the back of the eye front. Oops. Didn't mean to click that, meant to draw. Okay, so we're working our way from the back of the eye to the front. Okay, the first layer is going to be the photoreceptors, these blue guys right here. Okay, the photoreceptors are rods and cones, both of those together. The rods contain the pigment rhodopsin, and they are responsible for colorless vision in dim light. So if you are driving in the dark, your rods are going to be in high gear. They are picking up as much light information around you as you possibly can see. But you guys know when it gets dark, you can't see colors as well. Okay? And you don't see colors as well because you're not using your cones, you're using your rods instead. Okay? The cones, on the other hand, are for bright light and are responsible for sharp and color vision. And we'll spend some more time talking about our cones and how they work later. Okay, but both of these guys together, rods and cones, give you vision. They are going to be the ones that are taking photo information and changing it into an action potential. Okay, they are the receptor. All right. They then, once they start an action potential, they are going to send information to this next layer of cells, these red cells. Okay, the red cells are the bipolar cells. Okay? Then the bipolar cells will send the information to the ganglion cells. Okay? The axons of the ganglion cells. So these are your dendrites over here. They're picking up information. And then they're sending the information out these axons. The axons will join together, and they will eventually form the optic nerve. So that's where the information is leaving the eye and going back to the brain. Okay, but it all starts with these photoreceptors, bipolar cells, ganglion cells, and then their axons are the optic nerve. Okay? So if we look at our eyeball here, okay, so let's just pretend we're going to draw a little eye here. Okay, so the light comes in this way. It gets all the way back here to hit the photoreceptors. Then the photoreceptors send information to the bipolar cells and the ganglion cells. They send their information out on the axons. That eventually goes back this way out the optic nerve. Right? It makes sense. All right. So that is that. Here's just a different picture of what we just looked at. This is the same one as before. It's just blown up. And this is a picture of specifically the rods and cones. Okay, what I'm about to tell you is not important. Okay, don't worry about writing it down. But these photoreceptors do not reproduce. Okay, so you're not going to be able to replace them if they're damaged. But down here at the bottom, they sit in that pigmented epithelium, and the photoreceptor parts are down here at the bottom. Okay, and they're little discs. And so you'll have little discs that die, and they're phagocytized, but then a new disc will come down and take its place. Okay? So you're not replacing the photoreceptors, but these discs regenerate from the cell. Again, not important, but there it is. Okay. Okay, so talk with your neighbor. See if you can answer these two questions. We've already kind of talked about it. All right. So the bright white that you see is a combination of these two things. Okay? So for number one, when you're in the dark, what are you primarily using? Rods, because they're good for dark vision. And then when you step outside, you shift to cones. Okay, so you're shifting from a rod-driven visual pathway to a cone-driven visual pathway. Okay, so that's part of it. Secondly, when you go to the bright outside, are your pupils going to constrict or dilate? Constrict. But originally, at the beginning, they're still dilated until you get that massive input of bright light at the beginning. Okay? All right, so did I skip one, or am I good? I'm good. Okay. Your blind spot. Yeah, yeah, yeah.
But in a movie theater, when you're watching the movie and it's bright, with your eyes, but you're looking at the screen.
So you are looking at the screen that is bright. That is true. But the majority of the room and the majority of what's around you is dark. So, and then you're moving from there out into the hallway, which is dark. Thank you. All right. The rest. so it's right here, your optic disc, where that optic nerve leaves. Okay, so directly posterior to the lens is the macula lutea on the retina, and then a small patch, or it is a small patch that contains the fovea centralis that produces the most detailed image. Goodness gracious, I can't read anymore. Okay, so there's your macula lutea, most acute visual area. So this is where you want to focus things. Right over here, though, is your optic disc, and it's where you have no rods and cones. Okay, again, I hope you do this in lab, but if you haven't done it yet, you could do it with your notes because you got a picture here. Okay, so to see where your blind spot is. If you want to test your right eye, then you're going to cover your left eye, and then you're going to look, and this has to be something that's printed off that you can move, okay, or moving like forwards and backwards. Okay, but if you were going to test your right eye, cover your left eye, and you look at this spot, and then you move your paper forwards or backwards, or your head forwards and backwards, until this plus sign disappears. It'll just be like it doesn't exist anymore. Okay, and that spot where it disappears is where your blind spot is located. Okay, and then you can do it again for your other eye. If you do it for your other eye, you'll look at the plus sign, and the circle will be in your periphery, and you'll test it to see where it disappears. However, okay, so we know we have blind spots in both eyes, but the good thing is that we have two eyeballs, so that when this eye has a blind spot, this eye can see what's in that blind spot. Okay, and so you're not really missing information if you're looking with both eyes. Okay, if you lose vision though in one eye, this can become an issue because things will end up in your blind spot and you can't see them, and they'll be on your periphery. Okay. All right. Some more homeostatic imbalances. Glaucoma. So this one, if you've been to the eye doctor, they've done a test for glaucoma, and they hold this little thing up to your eye and they shoot air at it, and it's the worst thing ever. Okay. So what they're testing for is your intraocular pressure, or the pressure inside your eyeball. So when they shoot that little puff of air at it, it's measuring how far your eye moves in because of the air. Okay, it actually causes your eye to move. Okay, but if your pressure is too high, your eye doesn't move in when the air hits it. Okay, so what happens in glaucoma? The drainage of the aqueous humor is blocked. So remember, in the anterior portion of your eye, you have the liquid portion, and your aqueous humor is constantly being produced and constantly draining out. So you maintain a pretty steady amount of aqueous humor. But if something happens and your aqueous humor cannot drain out and it's still being produced, then you get a buildup of aqueous humor in that anterior chamber. Okay? And as it builds up, your pressure builds. And as the pressure builds in your anterior chamber, it pushes back on the posterior chamber, onto that vitreous humor. And the vitreous humor can put pressure on your retina and on your optic nerve. And we know from our previous discussion of nerves that pressure on nerves is not good, right? So, this may be your normal vision, but then as glaucoma happens, as your pressure increases, your field of vision will steadily decrease. If it goes on too long, you can lose the vision in the eye. So, treatments. You start out with some eye drops. Some miotics will increase the rate of aqueous humor drainage, so make it drain faster. There are also drops you can get to prevent the production of aqueous humor, or at least reduce it, so you're not making as much at the beginning. Then, if the eye drops are not successful, you can do surgery to get in there and drain out the aqueous humor. But if you catch it soon enough and are able to reduce the pressure, then you maintain your optic nerve. Okay, cataracts. Okay. When you dissect your cow, apparently you don't dissect. When you dissect your cow eyeball, the lens is going to be hard and opaque and thick and white. Okay, that's not what the lens looked like when it was a cow, but the preserving process makes it opaque. But if you have cataracts, then your lens gets hard and thick. Okay, that is cataracts. Okay, and when your lens gets hard and thick, it can't move to focus on things. And if it becomes opaque and cloudy, light can't get through it. If light can't get into your eyeball, you can't see anything. Right, you're blind. Okay, The lens thickens and hardens because of added inadequate nutrients to the deep lens fibers. So you get clumping of the proteins and cloudiness of the lens. So this is a problem, especially for diabetics who have circulation issues. You're not getting those nutrients into the lens that you need. Okay. So treatment. This one's really cool. Okay. So treatment for cataracts. What they can do is they can take a laser and they can shoot it at your lens and break it up into little pieces, and then take a little suction and suck out the lens, and then replace it with an artificial lens. Cool, right? Okay. Now, we are getting to the portion of the eyeball where we're looking at the formation of the image, how the image actually forms, and how you perceive image. That just blows my mind because it's just so crazy cool. Okay? So we're going to start talking about this today. We'll probably finish it on Wednesday. Okay? But here we go. Okay, so the iris has two sets of muscles. This is the same as what we saw in the other picture. It's just putting words to it. Okay, so you guys know these two, pupillary constrictor and pupillary dilator. Okay? What is new is this last bullet. Pupillary constriction in response to light is called the photopupillary reflex. This reflex includes the pupillary light reflex and the consensual light reflex. Okay, so if you take your eyes, as you do, and you cover up one like this, and you take a flashlight, and shine it in this eye, what's going to happen to this guy's pupil? Constrict. That is the pupillary light reflex. When you shine a light in your eye, it's going to constrict. Okay. The consensual reflex is that if I have my hand here and I shine a light, oh, you do have a flashlight. If I shine a light in this eye and the flashlight does not hit this eye still constricts. Okay? So the eyes are connected to each other. So if this pupil constricts, this pupil will also constrict. If this pupil constricts, this pupil will also constrict. Yeah. It's going to be something in that reflex pathway is messed up. Yeah. So the information isn't going to get from one side of the brain to the other. Yeah.
Is that why when you get a concussion at the end, they don't
Yeah, they're testing that light reflex. Yep. All right. So pupillary light reflexes. Okay. Image formation. So refraction. We aren't going to spend a lot of time talking about what refraction is. Yes. Yes. Yeah. Okay. So a pupillary constrictor is just a general term for the specific muscle. Okay. Refraction. We aren't going to spend a lot of time on the details of refraction and what causes light to refract. If you took physics, then you have drawn a lot of these diagrams, and they're really fun. It's one of my favorite parts of physics. But refraction is just the bending of light rays. And light rays bend when they enter a different medium. Okay? So when they change speeds, when they go from one media to another. You see this in a straw in a cup. Okay. Deborah figured out this at Thanksgiving while we were sitting and eating our turkey. She usually drinks out of a little opaque cup with a straw. But since everybody was drinking out of big fancy cups, because it was Thanksgiving, she asked if she could use my big cup with a straw that's clear, that has owls on it. So Deborah drank out of her big owl cup for Thanksgiving, and she could finally see the straw in the cup, right, because it's clear. And she said the straw is broken. And so we had a whole discussion over Thanksgiving about refractive indexes and how light can bend because it changes speed. That's great. My husband just looked at me like I was crazy. Okay. So this happens, the light bends as it enters the eye because it is moving from the air into tissue. So it has to go through the cornea, through the aqueous humor, and through the lens. Okay? So as the light enters the eye, it's first refracted by the cornea. It's bent a little bit. Not much, but just a little bit. The aqueous humor, though, does not greatly affect it. Its refractive index is about the same as the cornea. But then, when the light hits the lens, it's going to bend. Okay? And depending on the shape of the lens, determines how much the light bends. Okay. Now, these are beautiful pictures here, but I'm going to draw another one. Okay? So, if I draw right here a tree, beautiful tree, and then this is the lens of your eye. Now, first of all, I don't know why that tree is that close to your eye, but we'll just, skip over that part. When the light from the tree enters into this lens, it is going to cause it to bend, and it causes it to flip. Okay, that actually happens inside the lens, but it's okay. Okay, and what you end up with is a tree that looks like this. So, the light that comes into your eye gets flipped upside down before it hits your retina. So what your retina is seeing is an upside down picture of the world. It's crazy, right? And then your brain has to figure that out. It has to flip it back around to where things make sense. Okay? So that is, to me, the first layer of craziness that happens in your brain. Okay? Your brain has to flip it right side over. But that isn't just it. Okay, there's going to be some more stuff going on later. All right, the near response. So your eyes have to look at things that are up close to you, and they have to look at things that are far away. When you look at things that are far away, your eye is pretty relaxed. Okay, it's doing its thing, it's looking at things far away. But when you have to look and focus at something up close, we have a couple of things that has to happen. Okay, these things are called the near response. Okay, so this is what happens in your eye when you look at something up close. Okay, first of all, the lens has to change shape in order to bend the light to focus it on the retina. Okay? We're going to see a picture of this in a second, but the lens to see far away is flat. The lens to see something up close is bulging. Okay, it's a bulging lens. Okay. Secondly, I'm going to read this one to you, and then we're going to explain it, because it is a lot of words. Okay, so it says convergence of the eyes orients the visual axis of each eye towards the object in order to focus on the fovea centralis of each eye. Okay. So we know that the fovea centralis is where we want to focus things, right? That's the point of the highest visual acuity. Okay, and that means that I need to be staring at something directly in order for the light to come in and hit it at the very back of my eye, right? If I'm looking at something and it's over here and it comes in, it's going to hit over to the side somewhere. It's not going to hit on that fovea centralis that's in the middle. So, will you do something for me? Will you hold up your pen or pencil in front of you? Far away first. Okay, so stare at it. Now bring your pen closer to your nose. What happened to your eyes? They crossed. They converged. Okay, so that's all that bullet says. Is that when something gets closer, your eyes converge together to make sure that what you're looking at is going to hit the fovea centralis. All right. Next, number 3, constriction of the pupil adjusts the amount of light and reduces spherical aberration. So when you look at things that are up close, your pupil constricts. Okay. It does this to reduce the extra light that comes in. Extra light will cause this thing called spherical aberration, which just makes things blurry. Okay, and finally, the lens accommodates by means of the ciliary muscle, which we'll get to on the next slide. The closest an object can be and still be in focus is called the near point vision. This distance increases with age. So, again, if we are gonna look at something and see how close can you hold it to your face and still read it, I can probably get about right there. If I hold it any closer, it's gonna get blurry. Seen the old people though. You have to go like this to read it, right? That's because their near point, okay, their near point has gotten a lot bigger. They can't hold it as close and keep it in focus. Okay? You guys can probably hold the piece of paper closer to your face than I can because I'm older than you. Okay? So what happens over age is those ciliary muscles get weaker, your lens is not as flexible, and you're not able to bulge your lens as much. So you can't focus on things that are as close to your face. Okay? Alright. So, the near response in pictures. When you are looking at something that is far away, your lens is nice and flat right here. Okay, he is flat because the ciliary muscles are relaxed. Okay, and so he's pulling up, and the relaxed ciliary muscles will tighten. The book calls it the ciliary zonule, but we call it suspensory ligaments. Suspensory ligaments is the older term. Okay, but it makes those tight, and so they pull up on the lens and makes it flat. When you're looking at something up close, the ciliary muscles will contract, which will pull them down. That will loosen the suspensory ligaments and cause that lens to bulge out. Okay, so we're loosening the tension on the lens so he can go back to a little bulgy shape. Okay. All right. We are doing good, guys. Okay. Refraction problems and corrections. So some of us are born with eyes that do not see as well as others, and we have to wear glasses. So if you have perfect vision, you have an eyeball like this guy up here, the emmetropic eye. We could really say that you have a beautifully shaped eye. Okay? Beautifully shaped eyes that are nice and spherical give us good vision. However, if your eye is more like this football shape, then you are myopic or nearsighted. So you can see things that are close to you, but not things that are far away. And that's because with a longer eye, your lens is focusing the light at this plane right here. So that's where it's focused. Anything beyond that gets blurry. So in order to fix it, we add an extra lens on the front, a concave lens, which will move that focal point back to where it's actually focusing on the retina itself, and it'll make it clear. Okay? The opposite of that, hyperopic eye. You are farsighted, okay, so you can't see things up close. Your eye is more of a squished shape, it's a shortened eyeball. So the point of focus is here beyond the eye. So to fix that, you give it a convex lens, and it will move that focal point onto the retina, and then you can see clearly again. All right, so I believe, let's see, 23, I still have 2 minutes, guys. Right? Yeah, okay, one more time. Color vision. Okay, so cones. Color vision is made possible by 3 different cones, a set of cones that are named for their absorption peaks. So blue has a peak here at 400 nanometers. So it picks up blue light really well, but then some greens down here too. The green cone picks up in the green area, and the red cone picks up, some reds, and it picks up some greens and yellows. Okay, so with these 3 pigments together, we're able to pick up lots of different wavelengths. So you can see the different colors of the rainbow because they are activating those 3 different cones. It's kind of like all of our taste is made up with those, the umami, the sweet, the salt, and it's just a combination of those. Color is made up by the combination of red, green, blue. Okay? And so you've got cones spread out along the back of your eye. There's red cones, there's green cones, there's blue cones, and they're activated by the light that comes in. And somehow your brain takes the information from each of those cones and says, okay, there's a green light coming in right here, there's red light coming in here, there's blue light coming in here, and somehow makes an image out of it. This is crazy, guys. All right, y'all ready to go? I will see you guys on Wednesday. Make sure you're ready for your quiz. And then maybe y'all pray.